Continuing the discussion on development of microbiology in 19th century, that is golden era of microbiology from 1857 to 1954. The other field in which the major developments took place was developments in disease prevention. So the first part in that was discoveries and the inventions related to surgical antisepsis. So, this particular area in which several discoveries were made, that is, in relation to the prevention of infection or the prevention of disease, as well as the treatment of disease and treatment of infections. That means this particular area of the developments were in relation to the discovery or the synthesis of chemicals which prevents or which kills the different disease causing microorganisms. So first part in that is surgical antisepsis. Surgical means the surgery where the tissues are break open with scalpel or scissor for the treatment of certain disorders or diseases. That means an operation. Antisepsis. Sepsis means poisonous effect of a particular toxic compound, that is called as sepsis, means poisonous effect. Antisepsis means preventing the sepsis, preventing the toxic effect of that particular compound or microorganism. Now, what this surgical antisepsis means? Surgical antisepsis means prevention of sepsis, means prevention of infection or prevention of toxic effect during surgery. It is called as surgical antisepsis. So the person who contributed in this very significant work was Joseph Lister, an English surgeon. In 1860s, the deaths from infection following surgery were frequent. For example, 45% of Lister's patients died due to surgical infections. Means the rate of death due to the surgical infections was very high as compared to the deaths due to the infections, primary infections only. Lister used carbolic acid, which is called as phenol. So he tried to find out some way to prevent the infections which occurs during surgery. So whenever the tissues are break open, then these tissues are exposed to external environment and microorganisms get chance to enter into the tissues, and this is how it leads to the development of infection, the development of pus and other complications, and due to which the patients used to die. In order to search for a solution to prevent the deaths due to surgical infections, Lister found out a chemical, that is carbolic acid, which is phenol. So this phenol was already, it was known to kill bacteria. So what this Lister did, he used a dilute solution of this phenol, and then he soaked these surgical equipments like scalpel, scissor, pins, etc. etc. All these surgical equipments which are required to break open the tissues were first soaked into this phenol. Nowadays we call it as disinfection. He also used or adopted a method as it is seen in the picture to spray this phenolic solution in the operation theater or the operating room. So that resulted into the disinfection process and the killing of bacteria that were existing in that operating room. So with this technique, he got a huge success. He was able to prevent the deaths due to surgical operations, due to surgical infections. So, with the success of this technique, it was immediately accepted by other surgeons who also got remarkable results. So, in other words, he could prevent or he could stop the contamination of tissues due to the use of this disinfection technique by using phenol. Lister's findings were the origin of present-day aseptic technique that prevent infection. Aseptic technique means, if you remember, when we take any injection or in muscles, so usually physician or a doctor, he puts that alcohol swab onto your skin. So he cleans that skin part with the help of that alcohol swab or the spirit, that is to make that skin free from microorganisms. That is the aseptic technique. Then a variety of chemicals and physical agents are available today that can reduce the number of microorganisms in operating rooms, nurseries for premature infants. So it was a very significant contribution of Joseph Lister in relation to surgical antisepsis, and that was the beginning of disinfection technique or the aseptic techniques. Then the next part in the disease prevention is vaccination or immunization. Vaccination or immunization, it was introduced by Edward Jenner in 1798. Edward Jenner introduced the process of vaccination in 1798. There is an interesting history behind this particular discovery. So Edward Jenner, he immunized the people with smallpox, म्हणजे देवी. Edward Jenner observed that the boys working with cows, means the boys working with cows, cowboys, were not susceptible to smallpox disease. That means these boys were resistant, showing resistance to smallpox disease. So Edward Jenner tried to find out the reason why an ordinary boy develops smallpox disease. However, these cowboys are not getting smallpox disease. So he was trying to search for this reason. Edward Jenner tried to find the reason for this. He realized that there must be some material from these cows, and these cows were infected with a similar kind of viral disease, that is cowpox disease. So some material from these cows is getting transferred to these cowboys randomly because they were in close contact with these cows. So this material which these cowboys were receiving from these infected cows must be responsible for causing the resistance in these cowboys. So it was his proposal or it was his prediction that the boys, or it was his observation that the boys which were in close contact with these cows suffering from cowpox disease, and there must be some relationship between this close contact which was not seen in case of ordinary boys. Considering this particular observation, what he did, he vaccinated, or he took that cowpox material and he inoculated a boy, the first person in the world who was vaccinated. It was a boy named James Phipps. So he was vaccinated by inoculation of cowpox material. In the picture you can see the method of inoculation. You can see there is a scalpel or pin in the hands of Edward Jenner, and he is inoculating the cowpox material through a drop and by making scrapings. So a cut is being made or the scratches are being made by this needle or scalpel, and that was the method of inoculation that time. Nowadays we have a method of direct injection into muscles, so that was the old method. So what this Edward Jenner did, there was a disease called as cowpox disease which was there in the cowboys. So the preliminary symptom of this cowpox disease in these boys was development of blisters on the skin. So it was a mild form of disease. So what this Edward Jenner did, he took the scrapings from cowpox blisters found on a milkmaid and using a needle scratched the scrapings into the arms of James Phipps, who was an 8-year-old boy. Later on, after a few days, Phipps became slightly ill. That means he developed fever and his scratch turned bumpy. Very soon, Phipps recovered and was then exposed to smallpox. That is, he developed resistance against the smallpox disease. That is how this cowpox material, which was injected into the body of James Phipps, was responsible for the development of resistance. However, Edward Jenner, he was unable to explain the principle behind this development of resistance. That means he was unable to explain the principle of vaccination. However, this word was coined, that is vaccination. Vaxa means cow. So he used that cowpox material, and that is why the process is called as vaccination. Nowadays we call it as immunization also. The principle of vaccination was explained and demonstrated by Louis Pasteur. Louis Pasteur also worked on the germ theory of disease, and he also worked on the methods to prevent diseases in humans and animals. During his work on infectious diseases in 1880, he isolated a bacterium responsible for chicken cholera, a disease in chickens. He grew that bacterium in laboratory in the form of pure culture in a growth medium. Then, to prove the Koch's postulates, that is, to prove that he had really isolated the organism responsible for chicken cholera. So, if you remember, we have learned in the last lecture about Koch's postulates, that is in relation to the bacterial diseases. So Louis Pasteur was trying to prove that this bacterium is responsible for the production of chicken cholera in chickens. Louis Pasteur arranged a public demonstration. That means he called on public meeting where he was supposed to demonstrate his experiment. So he repeated his experiment in front of public, in which he took that frozen culture, means the culture, bacterial culture, which was stored in refrigerator for several weeks. So it was an old culture, but it was pure culture of that bacterium grown in the growth medium. So he inoculated these chickens with that old culture, and he was claiming that the chickens would become sick and die. Unfortunately, what happened? None of the chickens died, and Louis Pasteur's experiment failed in front of general and scientific public. These results puzzled Louis Pasteur, that he was not able to prove the Koch's postulates. Later on, Pasteur realized that accidentally he used the old culture of chicken cholera, which was stored in refrigerator, and this culture, old culture, was used for inoculation. So he concluded, or he predicted, that the chickens did not get sick or die because the culture was not fresh, or the culture was old, and maybe because of this long-term storage in refrigerator, these cultures would have lost their virulence, ability to produce disease, and therefore the chickens did not die, or they survived. That was the theory which was proposed by Louis Pasteur. In order to prove this theory, again he repeated the experiment, which is called as chicken cholera experiment. This experiment was conducted by using two groups of chickens, as we can see in the figure, left hand side group and right hand side group of chickens. Now the first group of chickens, these chickens were inoculated with pure culture of chicken cholera bacteria, which was stored in refrigerator for at least 8 weeks. Means this first group of chickens were inoculated with old culture of chicken cholera bacteria. Now we call that culture as attenuated culture, means which have lost their virulence. After inoculation, few weeks later, obviously the same thing happened, that these chickens remained healthy. Then he took the same chickens which were previously inoculated, and then he took another group of chickens which were not previously inoculated. Now he performed the next step of experiment using these two groups of chickens. On the left hand side, you can see this group of chickens which were previously inoculated. On right hand side, you can see this group of chickens which were not previously inoculated, and these chickens were previously inoculated. So again, next what he did, now this time he took pure culture, fresh pure culture of chicken cholera bacteria. This fresh virulent culture of chicken cholera bacteria, and then he inoculated that into this group as well as into this group of chickens. After inoculation, this group of chickens which were not previously inoculated, and now they're inoculated with fresh culture, they became sick and died. Whereas the second group of chickens which were re-inoculated with fresh culture remained healthy. So this is how the inoculation of pre-inoculation with old culture of bacteria, the same culture but the old culture of bacteria, it prevented the development of disease in these groups of chickens. So this experiment clearly proved that the inoculation of pure culture of old chicken cholera bacteria, it acted as a boost, and it was responsible for the development of resistance. However, this culture was non-virulent culture or a virulent culture. This one was a virulent culture which acted as a material to boost the immune system of these chickens. So after the boosting of the immune system, when these chickens were inoculated with the fresh culture, the body of these chickens was ready for the incoming infection. So because of the presence of already developed resistance power, these chickens could resist the virulent culture of this bacterium, and that is why they survived. This is the principle of vaccination. This means that this pure culture, it acted as a vaccine. So very soon Louis Pasteur found an explanation that bacteria lose the ability to produce disease, that is, they lose their virulence or could not become or could become avirulent if they were grown fixed for several weeks under refrigerated conditions. So therefore these bacteria could not produce the disease. but instead they were responsible for stimulating the resistance power or immune system of those chickens. But after inoculation in the body, it stimulates the host body to produce some substance. Hence, body remains prepared to fight against incoming infection by virulent bacteria. Thus, whenever the virulent organisms enter in body again, they are removed from the body immediately. Therefore, the chickens which were inoculated previously with old cultures survived after the re-inoculation of virulent bacteria. And nowadays, we know that the body's immune system, that is, the protective agency of body, is stimulated by first exposure to microorganisms. So this experiment of Louis Pasteur gave the complete explanation of the principle of vaccination, which was introduced by Edward Jenner. Pasteur called the avirulent cultures as vaccines and immunization as vaccination. Later, Louis Pasteur also worked on the development of a vaccine against development of rabies disease. We know that rabies disease is caused by the bites from the rabid animals like wolves, dogs, cats, bats, etc. etc. So next, Louis Pasteur prepared a vaccine against this rabies disease, which is also called as hydrophobia, which is being transferred from rabid animals to humans. So during that period, there were a lot of deaths of people, young people, old people, or the teenagers due to the bite of these rabid animals because no cure was available that time to this particular disease. Although the causative agent of this disease was not known, but Pasteur, as he worked in relation to the germ theory of disease, he proposed that this disease also involves an organism and microorganism. So Pasteur felt strongly that it is a microbial disease. There must be a microorganism involved in causation of this rabies disease. So considering this theory, Louis Pasteur, he collected saliva from a rabid animal. This is, you can see in the picture, this is a dog, and Louis Pasteur is collecting saliva with the help of a pipette from these rabid animal. He inoculated healthy rabbits with this saliva from rabid dogs. Very soon, these rabbits, they developed the disease and they died because of this infectious disease. Then, after their death, the rabbits were dissected to remove the brain. Now, why this brain was removed? Because Louis Pasteur, he observed the symptoms of this rabies disease in humans. So the symptoms were violating actions and abnormal actions of the people or the person who is suffering from rabies disease. And he strongly believed that When these abnormal actions comes out of any person, whenever there is an effect on brain, so he strongly believed that this causative agent must be affecting the central nervous system, and therefore he targeted the brain of this rabbits. So the brain was removed, dissected. It was dried for several days under aseptic conditions, and later on he obtained a powder. This powder was mixed with sterile liquid, and he prepared a suspension of that. Then this preparation was used as vaccine. So what this Louis Pasteur did, he inoculated this suspension into dogs, and he got amazing results that these dogs did not suffer from rabies disease. So that was his vital finding, outcome of very important experiment. After this particular experiment, there was a boy called as Joseph Mister. This boy was bitten by a rabid wolf, and his family requested Louis Pasteur to inoculate the child or to do some treatment to this child to protect him from development of rabies disease, which was a life threatening disease. Means that time death was for sure after the bite of rabid animals. However, Louis Pasteur was a chemist. He was not a physician or he was not a surgeon, and he was not accustomed to treating patients. Still, the family of Joseph Lister, they requested Louis Pasteur to do some kind of help. Louis Pasteur was worried. The worried Louis Pasteur inoculated the boy with the same material which was which was prepared by him from the rabbit's brain. So he inoculated that boy for several weeks, and in this case also he got the amazing results that the boy did not suffer from rabies disease and he did not die. That was a remarkable achievement of Louis Pasteur. Later on, several farmers, peasants, and several people used to come to Louis Pasteur to get inoculated after the bite from rabid animals. Likewise, a group of Russian peasants who were bitten by a rabid wolf, they approached Louis Pasteur, and Louis Pasteur inoculated them, and all these farmers, they did not suffer from rabies disease, and they got protection, and they did not die. The Caesar was very happy, the Russian Caesar, and he sent him 1 lakh francs as a reward money to Louis Pasteur. This money, along with their donations, other donations from all around the world, it was the beginning of the world famous Pasteur Institute of Paris in France. This is the Pasteur Institute nowadays, in front of which there is a statue of Louis Pasteur, who was the founder of this Pasteur Institute. And in front of this institute, there is one more statue which we can see here, that is in the honor of Louis Pasteur, development of rabies vaccine. You can see a boy, he is battling or fighting with this rabid wolf or dog. The same picture is seen here. Here also you can see a boy who is struggling with this rabid animal. So this is just a symbolic statue that Louis Pasteur's finding were significant enough, important enough to protect the people from getting suffered from rabies disease. That was a vital contribution of Louis Pasteur in microbiology.